Hey everyone. In this video, we're going to look at an overview of the process of DNA replication. Whether in prokaryotes or eukaryotes, DNA replication begins at designated locations on the chromosome, termed the origin of replication, indicated by the red mark here on the diagram. The chromosome may have just one origin, as is common on circular bacterial chromosomes, or it may have many, as is typical with linear eukaryotic chromosomes. When there are many origins, the same process will happen at each one simultaneously. The DNA strands will begin to be separated, opening up a replication bubble around the origin, with a replication fork at each end. Replication will proceed in both directions, moving away from the origin towards the replication fork. As the fork proceeds along the chromosome, parent strands will continue to separate, with new strands being synthesized to complement them until the entire chromosome has been replicated and separates into two daughter molecules, each comprised of one old and one new strand of DNA. Let's take a look at some of the major cellular components that are required for DNA replication. We, of course, need a template strand of DNA and a DNA polymerase enzyme to build the new strand of DNA. DNA polymerase requires a 3' hydroxyl group in order to bring in a new nucleotide. It can't build onto nothing. To get around this problem, the cell will use short RNA primers, synthesized by the enzyme primase, to provide the necessary hydroxyl group. Once all of this in place, DNA polymerase can progress down the template, building a new DNA strand from nucleotides within the cell. Now, there are a whole host of enzymes and other proteins that are required to make this process happen. As we mentioned, we first have to separate the DNA strands, and this is accomplished by an enzyme called helicase, which slides down the DNA, opening up the replication bubble as it goes. Opening up the double helix in one place and effectively pushing that twist down the molecule causes the DNA to over-twist further down, which causes strain on the DNA and can lead to a double-strand break. So, an enzyme called topoisomerase, which is also referred to as DNA gyrase, creates a nick in one strand to allow the strain to be relieved. Single-strand binding proteins can come in and stabilize the single-stranded DNA to prevent the bubble from closing back up again. As we mentioned a moment ago, primase will synthesize the short RNA primer that's required to provide the 3' hydroxyl group. This is usually around 10 nucleotides in length. Finally, DNA polymerase III can come in and start building the new DNA strand. Let's take a moment to quickly recap the structure of nucleotides and DNA as a whole, because this will impact how the DNA is replicated. Firstly, remember that a nucleotide is composed of a 5-carbon or pentose sugar, a phosphate group, and a nitrogenous base. In DNA, these are A, G, C, and T, while in RNA, the T is replaced with U. The carbons around the sugar are numbered, with the 1' being attached to the base and the 5' being attached to the phosphate. That 3' hydroxyl that we've been talking about is this one right here. And this will be used to form a phosphodiester bond to the phosphate of the incoming nucleotide. The structure of nucleotides is such that when the strands are assembled, they will be anti-parallel, meaning that one will be upside down relative to the other. So where you have the 5' end of one strand, it's partnered to the 3' end of the other strand. So why does it matter that DNA strands are anti-parallel? Well, it matters because DNA polymerase can only work in one direction. It can only add nucleotides to that 3' hydroxyl group that we saw. So it can only build DNA strands from 5' to 3'. Let's consider our replication bubble here, which, as you can see, is centered around the origin of replication. Since the strands are anti-parallel, the 5' end of one is partnered with the 3' end of the other. As DNA replication proceeds in both directions from the origin, we have two different scenarios going on. Firstly, we have the sections where the 3' end of the new strand is at the same end as the replication fork. These are referred to as the leading strands, and replication of these sections is relatively straightforward since everything is going in the same direction. The other sections, however, present more of a problem, since the 5' end of the new strand is at the same end as the replication fork. These require a more complex stepwise synthesis process, which we'll look at in more detail in another video. Thanks for watching, and please be sure to check out the videos on both replication of the leading and lagging strands.